Thanks, Heather. Good morning, ladies and gentlemen, and welcome to the CurePSP East Coast Conference on PSP, CBD, and related brain diseases. My name is Richard Zyne. I'm President and CEO of CurePSP, and since 1990, CurePSP has been the primary source of research and support for patients and families living with neurodegenerative brain diseases. First of all, I want to thank the Robert Wood Johnson University Hospital for its generosity in co-sponsoring this event in this wonderful space. And I'd also like to thank the PSP Research Foundation for its continued generosity in support of our research program, and all of the generous donors who continue to support our critical mission of programs and research. My purpose here this morning is to give you an overview of CurePSP and in particular our research program. Our vision is a world free of PSP, CBD, and related diseases. Our mission is to fund research, provide support for patients and their caregivers, educate healthcare professionals, and provide public awareness and advocacy on behalf of our patients. CurePSP is the only organization in the United States and Canada that focuses its resources primarily on PSP and atypical Parkinsonian diseases. People are always asking me about research. When will you find a cure? What benefits have come out of your research that will help me now? What causes PSP? What about the effects of heavy metals or pesticides? Do you use stem cells? What about transplanting healthy brain cells into sick brains? Yes, there is an urgency in our research, and every person who is suffering from PSP and CBD and the other diseases deserves a good answer. Good research is based on science, and it takes nothing for granted, and it assumes nothing at the outset, and it must always be shared with other scientists. At CurePSP, under the leadership of Dr. Lawrence Golby, CurePSP's research program has served as an incubator for innovative projects that have served as foundational work for clinical and therapeutic interventions. We promote research that helps generate an integrated understanding of PSP and related diseases at both the molecular and the cellular level. CurePSP provides grant support for the highest quality research and provides a forum for collaboration among researchers. Since 1997, the Foundation has awarded over 125 grants valued at more than $9 million. The research program has 4 basic goals. At the same time as we're trying to understand how these diseases originate and how they affect the brain and the body, we're also trying to identify the people who actually get the diseases and the genetic aspects and how the environment might also play a factor. Because it's always better to recognize and stop a disease before it can do harm, CurePSP wants to help develop diagnostic tests and biomarkers so that we might be able to see those diseases in action at an early stage and nip the diseases in the bud before they blossom into full-blown PSP and CBD. So our researchers want to develop a blood test or a spinal fluid test an X-ray or a CT scan to be able to see PSP when it's just beginning. And at the same time, we are striving to develop treatments for symptoms or medications to stop these diseases. Doing research is sometimes like looking for a child in a vast wilderness. And believe me, if our scientists knew exactly where to find that lost child, they would go to him immediately. So we have to begin at a starting point in this giant unknown area and identify the disease by what it appears to do to the body, such as symptoms and signs. And once we've given it a description, we begin to move in various directions, just like searching for a child in a large wilderness. And back in 1964, Drs. Steele, Richardson, and Olszewski did just that, and they gave a description and name to PSP, which is progressive supranuclear palsy. Well, sometimes the search is for those very basic causes, and maybe it is genes, and maybe it's environment, and maybe it's trauma, and maybe it's injury, or maybe it's actually a combination of all these factors. And at the same time, as we look for causes, we're searching for ways to help people while they are managing and suffering with their diseases. And we're looking for ways to detect these diseases when they can't be detected by the usual tests. And then there is the research to help people live a better quality of life by developing palliative treatments of symptoms to help people see better, to be less depressed, to have more energy, to be steadier on their feet, and possibly even to live longer. And of course, we're looking for the drug that will halt the disease in its tracks and even prevent it from happening. And the clinical trial is a vital part of this research. Sometimes we're successful, but other times medications either don't work or they're not tolerated well by our patients. CurePSP funds various types of research projects. The investigator-initiated research projects come directly from scientists. Researchers as well as clinicians come to us with their ideas to study a specific part of the puzzle. It might be an aspect of genetics, a study of one of the various forms of proteins that affect cell degeneration. It might be an exploration of a part of a cell, an epidemiological study, or even a treatment-oriented project. And then there is genetics. Since 2007, CurePSP has been a primary source of PSP genetics research in the world. The Charles D. Peebler Genetics Program has been instrumental in exploring new genetic components and aspects for PSP and CBD. The CurePSP Brain Bank at the Mayo Clinic has been instrumental in brain pathology research and is a prime source of brain tissue for the Peebler Genetics Program. And our research would be impossible without this invaluable resource. Our scientists share their discoveries with the world. Our annual research symposium enables scientists who are receiving funding from CurePSP to present their findings to our scientific advisory board and to share their ideas with scientists from around the world. Well, what have we learned after doing almost 20 years of research? Our research has taught us that PSP and CBD are underrecognized, underdiagnosed brain diseases that are often misdiagnosed as Parkinson's disease or Alzheimer's disease or even psychiatric diseases such as depression. The root causes of PSP and CBD are still unknown, but there may be both a genetic component as well as an environmental component for the disorders. And our research also holds insights into understanding and treating other atypical Parkinsonian diseases, as well as Parkinson's disease itself and Alzheimer's. Understanding brain proteins is essential to understanding why brain cells die. And a variant of a gene called tau is more common in persons with PSP than it is in the general population. This gene produces a protein that abnormally clumps in brain cells. An understanding of how these proteins clump and tangle in PSP and CBD has emerged during this period. And although tangles are found in other brain diseases, work on the tau gene indicates that PSP tangles differ from those in other diseases, such as Alzheimer's disease. Then there's alteration in cell energy, which occurs in PSP. This alteration is also found in cells located outside of the PSP patient's brain. So this suggests that changes associated with PSP may not be just limited to the brain. Various laboratory animals have been genetically engineered to develop PSP-like changes in the brain. These animal models have greatly accelerated the search for treatment for PSP in humans. And of course, there are genes other than the tau gene that may contribute to PSP. And that takes us to our genetics program. An understanding of the role of genes and what they play is critical. That's why we have spent so much effort and time and resources in this area over the past several years. We feel that there is an important interplay of genetics, molecular biology, environment, and toxins in the development of these diseases. New technology has enabled fast testing of brain tissue of thousands of samples of genetic markers covering every known gene. This technique involves scanning DNA from a huge number of patient samples. This powerful technology forms the foundation of the whole genome analysis, which you've probably heard about, or the WGA, which has recently been completed by Dr. Gerard Schellenberg at the University of Pennsylvania, and he's also the coordinator of our genetics consortium. The completion of the WGA is only the first step in the work of the genetics program. And phase 2 will study various proteins within the implicated genes that may enable scientists to correct defective biochemical pathways, which may help to prevent, slow, or even cure PSP. The consortium completed the WGA with over 2,000 patient samples. The results were 6 gene variants or defects that were much more common in the group of people with PSP. And later this month, the CurePSP Genetics Consortium will have its findings published in a major scientific journal, and the Foundation will promote the study and spread the message about our need to cure PSP. First, the genes that have been newly incriminated in the cause of PSP and CBD affect the production of tau protein. The next step is to determine just what this gene is doing wrong and find ways to fix it. Our scientists think that the newly found variant simply increases the amount of tau produced. And if this is confirmed, then clearing the excess tau from the cells could slow or even halt the progression of PSP. Second is the effect on cell vesicles, which are like tiny water balloons used for transporting chemicals around the brain cells. They're critical, and if they malfunction, the cells die. Two defects are in genes that help the brain package chemicals inside these vesicles. We need to find ways of compensating for these defects. A likely first step would be to remove these genes from an animal model, observe the results, and then attempt to correct them with drugs or gene therapy. Third is the gene defect on myelin, which is the insulating material around the long tubes called axons that grow between brain cells and which conduct electrical current. While loss of myelin is not known to be a feature of PSP, a defect in myelin could impair the function of brain cells. This observation creates a connection between PSP and multiple sclerosis, a disorder with myelin loss. Scientists will now look at myelin more closely in brain samples from PSP and CBD for clues to how this gene defect is working. Fourth are the two gene defects that impair the brain's ability to dispose of proteins. Such a malfunction is strongly suggested in Alzheimer's and in Parkinson's diseases and other neurodegenerative diseases, including PSP. The cause of the malfunction remains unclear in PSP. If not properly diagnosed, this lack of ability to dispose of this protein can become toxic to brain cells. We already have ways of improving brain cells' handling of misfolded proteins in laboratory animals, and will attempt to adapt techniques for practical use in human beings. And fifth is a diagnostic test for PSP and CBD that does not have to wait until a person develops symptoms. The accuracy of these gene variants as diagnostic markers must now be assessed. Conclusions. First, we are closer to understanding how genetics affects the development of PSP and CBD. And second, while there is probably a strong genetic component to PSP and related diseases, the effect of toxins and trauma and other environmental triggers is still not fully understood. We hope that the epidemiological studies that are now being done by Dr. Irene Litvan will soon provide some salient results. Third, there is hope that new pharmaceuticals now under study and clinical trials, such as Alon Therapeutics' Davunitide, may help to alleviate symptoms and improve the quality of life. Fourth, there is hope that continued treatment of neurological symptoms will improve patients' abilities to maintain a higher quality of life. And Dr. Golby will share some of his treatments with you this morning. There is hope that a cure or prevention may be achieved during the next decade. But there must be a sense of urgency to the needs of people with neurodegenerative brain disease such as PSP, and we must find the lost child in the wilderness. And frankly, we can't depend on the federal government to do this because they don't have any money. And I just got back from a conference at the NIH where they're really struggling. They're cutting back on their research. So we have to raise money from private sources to do what needs to be done. We cannot wait for the federal government to do it for us. And CurePSP is determined to see its vision of a world free of PSP and CBD come to fruition, and we must all work together to find a cure. Thank you very much. Okay. We're going to have a panel, and after a few of the other speakers, and I would like to now introduce our next speaker. Make sure I've got this. Our next speaker